So my name is Lana Vasung. I'm going to present a work titled Prenatal Development of Major Fiber Pathways in the Human Cerebrum. So I wanted to start with a small video that actually has been done on the postmortem MRIs from the Croatian Institute for Brain Research. So the cortex has been extracted at the Alan Evans lab, and what it actually shows that during prenatal development there are dramatic changes in the cortical surface. So what's even more interesting that these changes are not like changes that occur in cortical surface are there are even more dramatical changes that occur beneath the cortical surface. So this is mostly because fetal human brain is composed of different transient compartments and different transient zones. And one of these zones is actually a subplate zone, which has been proposed to be a waiting compartment for fibers that grow in the human cerebrum. What is interesting also to show that during the development, especially during the mid-gestation, there is almost half of the entire brain that is occupied by a subplate zone. And this growth in the volume of the subplate zone is followed by the decline in the volume of the subplate zone and a parallel increase in the volume of the intermediate zone, which is basically fetal white matter. So in general, these are all interesting informations, but what's even more interesting is actually how the spatiotemporal changes in thickness of the cortical plate and subplate zone vary across different developmental periods and across different regions of the human fetal brain. So it is also important to mention that the majority of the neurons have already reached the destination around 20 post-conceptual weeks, although there are some studies that indicate that this is ongoing until first year, although it's a smaller percentage. So one is normally bound to ask, how does the establishment of connectivity actually drives these changes, spatiotemporal changes in cortical surface and cortical or subplate thickness? So one of our objectives was to assess actually the spatiotemporal changes, spatiotemporal ingrowth and outgrowth of cortical fibers using HARDI. Basically, we wanted to replicate what has been known from histological studies from Croatia. So for this study from the lab of Emi Takahashi at Boston, we used 9 post-mortem human fetal brains, which is, I guess, for all of you a small number, but for us huge because it's almost impossible to get a fetal human brains. The age was between 15 and 30 post-conceptual weeks. We scanned them with 4.7 Tesla, high resolution, we used HARDI. Post-mortem MRIs were manually segmented in different zones: cortical plate, subplate, intermediate zone, subcortical structures, and proliferative zones. And these regions were used as regions of interest in order to try to detect different groups of fibers. We used TrackViz to reconstruct the different fiber bundles. So in terms of results, what we see is that during prenatal brain development, there is predominant radial coherence of the telencephalon. So what does that mean? Here you can see in green, these are fibers that stretch from cortical plate to subplate or intermediate zone. In yellow, you can see the fibers that stretch from proliferative zones and lower subplate zone. And in red, you can see fibers that stretch from cortical plate to the proliferative zones. What are the exact counterparts of these streamlines that are reconstructed? We are not sure. What we know from studies in histology that this is most likely a combination of radial glia and the fibers, cortical fibers that are still growing. During the mid-gestation, this radial coherence is predominant. So I guess we need to study brain microscale like more in detail in order to answer these questions. What happens with the projection fibers? So the paper is divided into radial coherence, projection fibers, associational, and callosal. Very early during the development, somewhere around 18 post-conceptual weeks, we were able to detect all the projection fibers. So you can see in, for example, in pink, you can see corticopontine, you can see Muratov bundle, thalamocortical fibers, tegmental fibers, and basal ganglia fibers. What is important to say is that during this period of development, these fibers don't reach the cortical plate. There are just few fibers that reach the cortical plate. Moving to the next period, somewhere around 24 post-conceptual weeks, you can see all the aforementioned fibers, but now these fibers are connecting cortex with all the subcortical structures. So in a way, using HARDI and a high-resolution diffusion tensor, we were able to replicate the histological findings, and we are able to say that this is actually the period when cortico-subcortical connectivity is established. And it's important to mention that this is the earliest period where you have premature birth, babies that are born prematurely that actually survive. So it's important to have this in mind. Another thing also important to mention, because we are using diffusion tensor here in combination a little bit with histology. For example, a construction of thalamocortical fibers, geniculocortical projection fibers, you can see that somewhere around 32 post-conceptual weeks, these fibers connect thalamus and cortex. However, if you use dye tracing, you can trace these fibers somewhere around this period. So with diffusion tensor, we are lagging a few weeks behind in terms of detecting establishment of connectivity. What happens with callosal fibers? Very early during the development, also 18 post-conceptual weeks, we were able to detect callosal fibers that enter cortical plate, but portion of them actually stop in the subplate zone. Going fast forward, around 32 post-conceptual weeks, we were also able to detect the callosal fibers that enter cortical plate, but then there is also a huge amount of fibers that stop in subplate. So these results are kind of in agreement with the postmortem histological studies of Patricia Gorman-Brakic that actually showed that there is protracted development of corpus callosum during prenatal development, which is followed by pruning and losing of callosal connections. What happens with association fibers? So we were very early during the development, we were able to detect different components of association fibers, like middle longitudinal fascicle, different fiber bundles that connect different cortical regions, inferior front occipital fascicle, vertical occipital fascicle. Within the next period, we have been able to detect these long association pathways, even in the bigger volume, they were more voluminous. And lastly, around 30 post-conceptual weeks, you can clearly see outgrowth of all of these long associational pathways with the outgrowth of short cortical U fibers resembling. So this is actually the periods where this cortical surface, there is exponential increase in cortical surface and also cortical volume. So it might be relevant for the establishment of connectivity and the maturation of different cortical areas. Last thing that we did is actually we tried to map where, combining histology and MRI, where these specific fibers pass within transient fetal zones. This is just one example. So here you can see that the callosal fibers and different projectional and associational fibers actually pass close to the ventricles. And these are the regions that are most commonly injured in the prematurely born children, periventricular leukomalacia, which in a way explains the palette of neurological deficits these children display, from cognitive to sensory motor. So it's important to have it in mind. So as a summary, during the mid-fetal period, we see establishment of the projection pathways, development of the callosal pathways, and the radial coherence of telencephalon. During early preterm period, we have peak of radial coherence of the telencephalon. Projection fibers actually connect the cortex with the subcortical structures. Callosal and association fibers are still growing. And lastly, during late preterm period, we see projection fibers again that are connecting cortex with subcortical structures and prolonged protracted development of the associational and callosal fibers with the diffusion. What is the importance of a study that we are trying to assess? This is just the first step towards the questions that we are trying to ask, is actually how this establishment of connectivity, which is spatial regionally dirigated by different gene expression, how does this affect the maturation of the cortex, dendritic arborizations, synaptogenesis, changes in cortical thickness or cortical surface? And secondly, how do these type of studies can be linked with studies in connectomics. For example, 2 years ago we did a study where we showed that children born prematurely actually had weaker connections within cortico-basal ganglia thalamocortical loop, and they also display palette of neurodevelopmental deficits. So if you go to histological picture, injury in this one specific region of the brain could produce like different neurological deficits. So lastly, as a conclusion, we showed the correlation between histology and HARDI that's in the paper. We could detect sequential development of fiber systems. There's special relationship within transient fetal zones and their geometric properties. Thank you.